In this video, we'll be talking about proteins. Proteins are large biological macromolecules that can be pretty varying in size. They're all made up of amino acids, but the number of amino acids that go into making a protein can vary from a couple dozen to tens of thousands. So proteins are polymers, and the monomers that make them up are amino acids. Here we're looking at the generalized structure of an amino acid. All amino acids have this central carbon, and attached to that carbon you have a hydrogen, you have an amino group, that's this functional group, and you have a carboxyl group, that's this COOH group. What we really want to focus on when we're talking about the different amino acids is this side chain, the R group. This is what's different between different types of amino acids, and there are about 20 amino acids. Just like in the English language, we have 26 letters that can make an enormous variety of words and sentences and essays and books. Out of 20 amino acids, we get the enormous variety of proteins that we see in the human body and in other animals. And each amino acid has a different R group. That central carbon is the same, the amino group is the same, the carboxyl group is the same. It's the R group that's different between the 20 amino acids. Here I'm showing you the chemical structures of the 20 different amino acids. I didn't include this in your notes because I don't want you to think that I expect you to memorize the structures. I just have them here to illustrate, to show you the variation that we have. So if you take a look at every single one of these chemical structures, you see that they all have the central carbon with the hydrogen, carboxyl group, and the amino group around it. And then highlighted in blue here you have the different R groups. And notice that the R groups fall into different categories. So here you have nonpolar R groups. Here you have polar R groups, ones that are going to be more hydrophilic, these are going to be more hydrophobic. You have R groups with a positive charge, you have R groups with a negative charge, you have R groups with big carbon rings on them. There's a huge variation in both size of the R group, such as this one where the R group is just another hydrogen, and say this one, tryptophan, with 2 carbon rings, and differences in their chemical properties. Now, in the process of making a protein, amino acids join together, and the process by which they join together is called dehydration synthesis. So when you have 2 amino acids joined together, the carboxyl group binds to the amino group, and you lose, as those 2 join together, you lose a molecule of water. The bond that's formed is called a peptide bond between that nitrogen and the carbon of the carboxyl group. That's called a peptide bond, and that's why sometimes you may hear the term polypeptide used to refer to a protein or a fragment of protein. When you have tens to hundreds to thousands to tens of thousands of amino acids all strung together, this string of amino acids folds up on itself and takes on a very important 3-dimensional structure. And it's this 3-dimensional structure of a protein that determines what that protein does. Now, I mentioned that amino acids can have huge variation in size and chemical properties of their R group. Just to illustrate that, I'm showing you a more visual, more 3-dimensional model of 2 amino acids. Glycine, which has a very small R group, just a single hydrogen, and lysine, which has a much larger R group. So the lysine is going to take up a lot more space. It's going to have a lot different chemical characteristics from glycine. And if you think back to the previous slide where we showed the three-dimensional structure of a protein, the way that it folds into that three-dimensional structure, the way that it takes on that three-dimensional structure, depends on the characteristics of these R groups. If you have an amino acid that has an enormous R group, like lysine, that protein is going to fold in a slightly different way than if you just have a tiny R group. Whether that amino acid has a hydrophobic or a hydrophilic R group also can be important, because remember, everything that's taking place inside and outside your cells is in a watery environment. So in a watery environment, you're going to tend to have hydrophobic amino acids being found in the center of the proteins, that they're kind of hiding away from the water. Hydrophilic amino acids are more likely to be found kind of outside on the boundaries of the protein, where they can interact with the water. Now, there are different levels at which we can talk about a protein's structure. It's ultimately the complete three-dimensional structure that determines how the protein functions, but different aspects of protein structure go into making up that complete three-dimensional structure. And so we talk about there being 4 levels. And the first level is the primary structure, and that's just the chain of amino acids. So that's just the sequence of amino acids. If I wrote out, say, lysine, glycine, tryptophan, tyrosine, that's the primary structure of a protein, just what amino acids and in what order. The next level of protein structure up from that, as those amino acids kind of curl around and interact with each other, you have the secondary structure. And the secondary structure, there are really 2 main types of structures that you find at this level, and that's the alpha helix. And helix, remember, is just a fancy way of saying spiral. So sometimes that chain of amino acids can kind of spiral around itself. Or you can have what's called the pleated sheet, or sometimes the beta pleated sheet, and that's where that structure just folds back on itself. You can also have regions of a protein with not a well-defined secondary structure, where it's just a little bit free-form. Next, you have the tertiary structure. In tertiary structure, this is the 3D shape of a single chain of amino acids. I'm going to abbreviate amino acids AA. So you take a single chain of amino acids, when it takes on its full 3-dimensional shape, that's the tertiary structure. Quaternary structure is when you have 2 or more chains of amino acids interacting. And this is showing hemoglobin as an example, because hemoglobin has 4 amino acid chains that actually come together and interact to make a complete functional hemoglobin. Not all proteins have quaternary structure. Some proteins are complete at the tertiary structure. They're just made up of a single amino acid chain. But many proteins, like hemoglobin, do have multiple chains of amino acids folding together to give a protein its function. Now, the 3-dimensional structure of a protein is absolutely critical. Everything that a protein can do depends on how it interacts with other molecules in the cell. And how it interacts with other molecules is determined by its 3-dimensional shape. So, if a protein loses its 3-dimensional shape, it basically loses its function. And this loss of 3-dimensional shape is called denaturation. And there are a number of ways that it can happen. Excessive heat, if you have too high of a salt concentration, or if you have too low of a pH, or sometimes too high of a pH. These can all interfere with protein structure. you're all familiar with denaturation, although you might not necessarily think about it. But when you are cooking an egg, applying the heat in the frying pan, that's causing denaturation of all the proteins in that egg, and as those proteins denature, they come out of solution and they solidify, making the hard fried egg that many people love. Now, obviously, we don't fry our cells under normal conditions, but there are conditions where denaturation does change how things behave in the body. It's thought that muscle fatigue, when you're exercising at high levels, causes some changes in pH in muscle cells, and that might be a contributor to muscle fatigue. It's a reason why excessively high temperatures, excessively high fevers can be dangerous, because the proteins in your brain don't operate as effectively at excessively high temperatures. And so you can get things like seizures at excessively high fevers. So denaturation plays a role in cooking and in normal body functioning. All right. After studying this video, you should be able to answer these questions. Talk about the relationship between proteins and amino acids, describe the structure of an amino acid, explain the 4 levels of protein structure, and talk a little bit about both what denaturation is and why it's important.